Okay, so now we're going to talk about the phases of mitosis in animal cells. So just keep in mind that mitosis is often described as having 4 or 5 distinct stages, but it's important to remember that mitosis is actually a continuous process. So one step is flowing into the next. We do not have it just stop at one of the phases and then wait for something else to happen in order for it to move on to the next phases. It happens continuously. Just have a general idea of what's happening in each one of these phases. So we've talked about interphase before. So this is before mitosis even begins, and it's when the cell is busy preparing for division. So we have our cytoplasmic contents that are going to be duplicated, and we have DNA replication occurring inside of the nucleus. So even though each chromosome now consists of 2 sister chromatids, they still aren't really visible in our microscope. We also have a pair of centrosomes forming and getting into position outside of the nucleus. And these are just structures that will play a key role in cell division later on. Next, we have prophase. So this is where things start to become visible. We have our chromosomes beginning to condense, and now they're becoming thicker and easier to see. And each now consists of 2 sister chromatids that are joined at a centromere. While that's happening, we have the spindle apparatus that's starting to form between the 2 separating chromosomes. Remember, the spindle apparatus is just a network of microtubules that are going to assist in the movement of these chromosomes. So as prophase continues, the nucleolus is going to disappear and our nuclear envelope is going to break down, freeing those chromosomes into the cytoplasm. We have spindle fibers from both poles of the cell that are going to reach out and attach to chromosomes at these special protein structures near the centromeres that are called kinetochores. So as this connection forms, the chromosomes may wiggle a bit. They can move slightly towards one pole and then the other as they adjust to the pulling forces from both sides. In metaphase, the chromosomes are going to line themselves along the spindle equator, which is also called the metaphase plate, basically the middle of each cell. Each chromosome is going to be perfectly positioned with spindle fibers from opposite poles attached to either sides. We also have some other spindle fibers that aren't attached to chromosomes, and they're going to stretch beyond the equator and they're going to overlap. And these are just really there to help stabilize the structure. So now we can move on to anaphase, and this is where the real action is really going to happen. The sister chromatids are going to separate at the centromere, and they're going to become an individual daughter chromosome. These daughter chromosomes are going to be pulled to opposite poles of the cell as they're attached to the spindle fibers, and the spindle fibers begin to shorten. At the same time, the spindle poles themselves are going to move farther apart, because the unattached spindle fibers are going to begin to slide past each other, helping kind of stretch the cell and contribute to the separation of chromosomes. Then we have telophase and cytokinesis. So in telophase, the spindle starts to break down and we have to reform the nuclear envelope around the 2 sets of daughter chromosomes. Each of the resulting nuclei is going to contain the same number and types of chromosomes as the original parent cell. So we might still have remnants of the spindle fibers that may still be visible between the 2 nuclei, but the cell is still clearly on its way to becoming 2 individual cells. At this point, we can start cytokinesis, which is the actual division of cytoplasm. And this process is going to start during telophase. Some texts say it starts at anaphase, and it's going to continue until the cell physically splits into 2 distinct daughter cells. And each new cell is going to be genetically identical to the parent cell and to each other. So just kind of a quick recap. So DNA replication happens before mitosis during the S phase of interphase. Each chromosome enters into mitosis as 2 sister chromatids that are joined at a centromere. The stages of mitosis are PMAT, prophase, metaphase, anaphase, and telophase, and they are part of a fluid and continuous process. Then we have cytokinesis, which is also part of the mitotic phase, which is where we're physically separating the 2 new cells, usually follows mitosis, starts during anaphase or telophase. In diagrams, chromosomes from different parents, like I mentioned, may be color-coded, usually red and blue, to show their origin, but the process works the same regardless.